Alright, hey guys, so for this week we went over a variety of topics, and of them being pleiotropy versus epistasis. Alright, so what is pleiotropy? Pleiotropy occurs when one gene affects multiple traits. So here we have, for example, let's say we have gene 1. It is going to affect multiple traits. This is going to affect trait A, as well as trait B. And an example of that would be Marfan syndrome, in which it's caused by autosomal recessive mutation, in which it causes a person to have long arms, long fingers, and extended height. Epistasis occurs when more than one gene influences only one trait. So here we have, for example, gene 1 and gene 2, and both of them are going in to affect trait A. And an example of that would be Labrador coat color for dogs. In the case that a Labrador had a dominant B allele and a dominant E allele, the color of the organism would be black. In case it had two recessive B alleles and one dominant E allele, the color of it would be brown. In case that it had two recessive E alleles, regardless of what the other B alleles may be, whether they be dominant, recessive, the individual is going to turn out yellow. So these two little e's mask the effects of the B locus. So let's say that there were two individuals that were white. Now both of them have different mutations for those white colors. Say for this individual right here, this white individual, he has two recessive B's which code for a white color. Let's say for this individual right here, he has two recessive A's, and this codes for the production of pigment. So two recessive A's means that no pigment will be produced, so the organism turns out white. So they both have the same phenotype, just different mutations affecting the phenotypes. Now, if we were to cross both of these individuals, they would become heterozygous at each of the loci, meaning that the original or the wild type gene or the wild type phenotype would be expressed. Let's say the wild type was orange. So what happens then is complementation. What's complementation? Complementation occurs when two strains of an organism with different homozygous recessive mutations that produce the same mutant phenotype have offspring that express the wild type phenotype. So another example would be if we have the B allele right here. A big B codes for a wild type color. Two little b's code for a white color. The big A codes for pigment, the production of pigment. Two little a's code for no pigment, which ends up being albino or white. If we were to cross both of these strains of organisms together with the different mutations, though they have the same phenotypes, we would get an individual, or we would get an F1 progeny being all heterozygous, carrying at least one big allele for each of the loci, restoring the wild type allele, or the wild type phenotype. So in this case, complementation occurs since two mutants restored the wild type phenotype. How would a complementation test be used to determine a phenotype at the same gene? Alright, so let's say that there was a mutation at the same gene, at one specific gene. Let's say it's gene B. This codes for a blue color. If we were to cross an individual, little b crossed with little b, we would get all little b's, which means that all the individuals would be blue. In this case, complementation did not occur because it did not restore a wild type phenotype, and also complementation occurs between two mutant state strains of organisms with different homozygous recessive mutations. These are the same homozygous recessive mutations. So complementation would not occur. Here's another example right here, in which you have the same gene. You're crossing two black mice with the same genotype. You end up with F1 progeny, the same genotype, same phenotype. So the phenotype is the same as the parents. It does not complement. Going on to population genetics. Now here we're given a graph. On the y-axis, we have the frequency of allele little a. On the x-axis, we have the generations that pass by, going up to 1,000. Right off the bat, what does this tell us? So this right here is telling us the frequency of this allele, little a allele, in the population over time. We can see that for the red trajectory, population, or the frequency of this allele, decreases heavily over the past, in span of about 100 generations. Whereas for the B trajectory, it's slowly going down. The frequency of the allele is slowly decreasing. So right off the bat, this is telling us that this allele is decreasing. So what does that mean? Well, for an allele to decrease, it must be deleterious or non-beneficial. Natural selection is only going to act upon the most beneficial alleles to make them the highest frequency. The fitness for allele little a seems to be relatively low for the A trajectory, and also for the B trajectory, it's also decreasing, meaning that it has negative fitness effects on the organism, meaning that it's not the most beneficial allele in the population. So, in turn, natural selection is going to act against this allele, and it's going to decrease. So that is why the frequency of little a allele is decreasing. Alright, if the allele is decreasing at a far greater extent for the red trajectory compared to the blue trajectory, what does that mean? Alright, so let's say that this red trajectory right here is one type of population of the same species. Blue trajectory, a different population for the same species. What does that mean? Why is this decreasing at a further or to a greater extent as compared to the blue population? Well, that would tell us that this allele has greater effects on this population as compared to this population. So this allele is affecting this population more heavily and stronger as compared to this population. Alright, now let's say we were given a chart, and it's asking us which genotype is most fit, which allele is most fit, which allele is dominant, what outcome do you expect? Alright, so right off the bat we have the different genotypes right here. We have big A, homozygous dominant, the heterozygous, and the homozygous recessive. For the first row, we see that the homozygous dominant has the highest fitness you can have. So these numbers right here represent fitness. The higher the number, the higher the level of fitness. 1 being the highest or equaling to 100%. So here we have 100% fitness. Heterozygous has 100% fitness. Homozygous recessive has 0% fitness. So the fittest genotypes would be big A, little a, since they have the highest fitness and have equal fitness. The fittest allele would be homozygous, would be the dominant allele, or the big A allele, since whenever an individual has that allele, they have the highest fitness. The dominant allele is the big A allele, since we see that the heterozygous right here has the same fitness as the homozygous dominant. And we see that the homozygous recessive individual has a fitness of 0. So that means that the big A allele is masking the effects of the little a allele. In the long term, what's going to happen is the allele big A is going to fixate to 100%. So it's going to reach 100% frequency in the population in the long term. For column 2, or for row 2, we see that the most fit genotype would be the homozygous recessive. They have the highest genotype. So, or they have the highest fitness levels. So out of all of the different genotypes, we see that any of them containing a big A allele has a lower fitness than the homozygous recessive, meaning that the big A allele is decreasing the fitness of the individual. So the fittest allele would be the little a. The dominant allele would be the big A, since the heterozygous individual has the same fitness as the homozygous dominant. So it's masking the effects of the little a allele. In the long term, little a is going to try to reach fixation or reach 100% in the population, 100% frequency. For the third row, we see that the homozygous dominant individual has the highest fitness, followed by the heterozygous individuals, which are followed by the homozygous recessive individuals. So the fittest genotype would be the genotype with the highest fitness, and that is the homozygous dominant. The fittest allele would be the big A, since homozygous dominant has the highest fitness, homozygous recessive has the lowest fitness, and the intermediate has an intermediate between both of the fitnesses. So the dominant allele in this case would be both of them. They would be codominant. And codominance is when both alleles are equally expressed. So an example of that would be if you had red and white flowers and you were to cross them together, you would end up with a hybrid or a mixture of both red and white on a flower. So you'd have like perhaps a white leaf with like red dots all over it, in which they would both be expressed in the phenotype. But since the most fit allele is the big A allele, selection is going to act on this allele and try to increase it in frequency of the population. So in the long term, the most fit allele, the big A, is going to try to reach fixation or reach 100%. For the fourth row, we see that the highest fitness is the heterozygous individual. So the fittest genotype would be the heterozygote. Looking at both the homozygous recessive and the homozygous dominant, we see that the homozygous recessive has a higher fitness as compared to the homozygous dominant, meaning that the most fit allele would be the recessive allele, which is little a. In this case, what would be the dominant allele? It would be overdominant, meaning that the heterozygote has a more extreme phenotype compared to the homozygotes. And an example of that would be sickle cell, or individuals with sickle cell anemia. So sickle cell anemia is a disease or a condition in which an individual has two recessive alleles, and that causes a person to have sickle cell anemia. However, for individuals living in areas with high malaria, you'll find that a heterozygote has the greatest fitness, or he has more of a resistance to malaria as compared to the homozygous dominant and the homozygous recessive individuals. And overdominance is a bit confusing, so just understand that it has a greater effect on the fitness of an individual, or a greater phenotypic effect than both of the homozygotes. So for example, you'll have, just an example, let's say we had two individuals, little a, and this codes for a gene that codes for, let's say, horn length. The greater the horn length or the longer it is, the more beneficial it is for the individual. Let's say this codes for 6 inches. Now let's say we have a homozygous dominant individual, and that codes for 8 inches horn length. The heterozygote would, for example, have 10-inch horn length instead. So we'd have a more extreme phenotype as compared to the homozygous individuals. So that's an example of overdominance. In the long term, what would happen is balanced polymorphism. And what that means is both of the alleles are favored to the same extent, meaning that they're both going to be fixated to a similar degree in the long term. So instead of reaching, for example, 100% fixation at allele A, both of them are going to try to reach a high level of fixation because the most beneficial genotype would be the heterozygous individuals. All right, in the first row, why would it take longer than you might expect for big A to fix in populations? For the first row, the reason it would take longer for the big A allele to fixate in this population would be because the homozygous dominant and the heterozygous individuals share the same fitness. So the heterozygotes mask the little a allele. In the second row, what pattern allows you to infer that the less fit allele is the dominant allele? Well, the pattern that allows us to infer that is because the homozygous recessive individuals have the highest fitness as compared to the homozygous dominant and the heterozygous individuals. The heterozygous individuals show us that with the addition of the dominant allele, the fitness decreases, whereas the homozygous recessive individuals show us that those individuals have the highest fitness. Going on to lethality. Alright, so lethality essentially means that an individual carrying specific copies of a lethal trait is going to die. So there's two types of lethal alleles. You have dominant lethal and you have recessive lethal. Dominant lethal occurs when inheritance by one copy of the mutated allele, either a homozygote or a heterozygote, will result in the death of for the organism that carries it. Recessive lethal allele means that the inheritance of both alleles will result in death of the organism. Alright, so here we have an example. In foxes, two alleles of a single gene, big P and little p, may result in lethality, big P, platinum coat, big P, or silver coat, little p. What ratio is obtained when platinum foxes are interbred? Is the big P allele behaving dominantly or recessively and causing lethality? Or B, platinum coat? Alright, so right off the bat, we're going to be crossing two foxes that are platinum. The platinum genotype would be big P, little p, crossing that with another platinum individual, big P, little p. So if we were to do a Punnett square, we would get 1 out of 4 of the progeny having big P, 2 out of 4 of them having big P, and 1 out of 4 of them having little p. Now, lethality occurs when there are two big P's together. So this right here is lethal, meaning that this organism is going to die, it's dead. So we would have to fix our ratios. So we would not include this in the total ratio. So now we have to make a new ratio for the progeny. So now we have a total of 3 boxes. We have 2 out of 3 of them having the big P, little p allele, and 1 out of 3 of them having the little p and little p allele. Alright, so this is the ratio that would be obtained if these two foxes were interbred. Alright, is the big P allele behaving dominantly or recessively and causing lethality? So, in order for it to be lethal, you have to have 2 copies of this allele, meaning that it is behaving in a recessive manner. Because recessive lethal alleles occur when inheritance of both alleles will result in the death of the organism. B. Is it behaving dominantly and causing B, platinum coat? So the inheritance of one of the big P alleles will change the phenotype of the organism. So 2 little p's will result in a silver color. 1 big P will change the phenotype into a platinum color. So it is behaving dominantly since it's masking the effects of the other allele. Alright, going on to blood types. Alright, so in humans, there are 4 specific blood types. There's blood type A, blood type B, blood type AB, and blood type O. We have 3 total alleles, AI, IA, IB, and little I. Both of these alleles behave codominantly, so they're both equally expressed. This allele right here behaves recessively. So in order to get blood type A, you have to have 2 IA alleles or 1 IA with a little I. In order to have blood type B, you have to have 2 IBs or IB and little I. In order to have blood type AB, since they both behave codominantly, they're both equally expressed, you have to have both of the alleles for I and B. So it'd be IA and IB. In order to have blood type O, that is the recessive genotype, so you'd have to have 2 little i's. Also, if there was a question asking about Hardy-Weinberg for blood types, there was a specific equation you have to know. It's different from the traditional Hardy-Weinberg equation. So the Hardy-Weinberg equation, as we know, it's a way to determine the frequency of the genotypes and alleles in a population, assuming that a variety of conditions are met and no evolution is occurring. So for the genotype equation, you would have p squared plus q squared plus r squared plus 2pq plus 2pr plus 2qr equals 1. For the allele frequency equation, you would have p plus q plus r equals 1. And remember, these are all in frequencies, so they're all in decimals. Going back onto epistasis, here's a question. In addition to independently assorting C, little c allele is recessive to big C and prevents the mice from making any fur pigment at all, resulting in albino mice, regardless of the genotype at the agouti locus. 2 mice with the genotype AY, little a, big C, little c are crossed. What fraction of the progeny will have the yellow coat color phenotype? Alright, so here we have 2 genes which are impacting the phenotype of an individual. Alright, we have AY and little a. Now, AY is a recessive lethal allele, so it will result in lethality. AY, little a, will result in a yellow mouse color. Little a will result in a black color. Big C and any other allele will result in the making of pigments. Little c will result in no production of pigment. So let's say we had 2 individuals with the same genotype, AY little a, big C little c, crossed together. What fraction of the progeny will have the yellow coat phenotype? Alright, so let's make a Punnett square for both of the alleles. Here we have the A allele, here we have the C allele, the C gene. We have AY, little a, AY, little a. Alright, remember, AY causes lethality. We don't include that in the final ratio. So we have 2 out of 3 being AY, little a. 1 out of 3 being little a. If we do the C genes, the C gene, we have big C, little c, big C, little c, little c, little c. 1 out of 4, big C, big C. 2 out of 4, big C, little c. 1 out of 4, little c, little c. Little c, 2 little c's produce no pigment at all. Having at least one big C will result in the production of pigment, so both of these have pigments. So the ratio would be 3 out of 4 for pigment. In order to see the fraction of the progeny that will have the yellow coat color, we simply multiply the ratio for the individuals which can have the yellow coat color, which is 2 out of 3. We multiply that with the ratio of being able to produce pigment, which is 3 out of 4. And we get 6 out of 12, or 0.5. Alright, going on to sex linkage. What is sex linkage? Well, as the name suggests, there are genes located on sex chromosomes. Where am I going? Darn it. Alright, if a gene is located on the Y chromosome, only males can have it. If a gene is located on X chromosomes, males get only 1 copy, and females get 2. And this is specifically for humans. For some species, different chromosomes are used to determine sex. For birds, you have the ZZ, ZW, in which ZZ will produce a male, a ZW will produce a female. Alright, now let's say we had a husband and a wife with normal vision. Their fathers both have red-green color blindness, an inherited X-linked recessive condition. What is the probability that the first child will be A, a normal son, B, a normal daughter, C, a colorblind son, or D, a colorblind daughter? Alright, so right off the bat, we know that we have a husband and a wife, and they both have normal vision. And this trait is an X-linked recessive condition. Females can only pass down X chromosomes, whereas males can pass down both an X and a Y chromosome. So the husband, his genotype would be XY. The female, her genotype would be XX for the sex chromosomes. Now, since the husband does not have any, it means that he inherited a working X chromosome, meaning that he has the genotype XY. Let's say XC stands for the colorblind chromosome. The chromosome that has the condition, or the mutated allele, or the recessive allele. Alright, the mother, she inherited, since she has normal vision, she inherited 1 normal copy from her mother. Since her father was colorblind, he only had 1 X allele, and she gets her other X chromosome from her father, meaning that she received the recessive X allele. So she's a carrier for the disease, but she does not express the condition since she has 1 working X chromosome. Now, if we were to cross both of these individuals, what's the probability that their offspring will either have color blindness or no color blindness? So if we were to do a Punnett square, here we have an XY for the father, we have an X and an XC for the mother. Alright, so what we end up with is 1/4 of them having XX, 1/4 having XY, 1/4 having XC, 1/4 having XCY. So 3 out of 4 of these individuals have no color blindness. Now this individual right here, this female, she is carrying the allele for color blindness, but since she has 1 dominant allele, that is not going to be expressed. So we have a female here, no color blindness, a male, no color blindness, another female, no color blindness, and now we have a male that does have color blindness since he has one of the recessive chromosomes. So what's the probability of having a normal son? We see that there's a 1/4 probability or ratio of having a normal son, so it'd be 1 out of 4, or 0.25, having a normal daughter. Here we have two individuals that are females, that are daughters, with normal vision. Granted, one of them is a carrier, yet she still has normal vision. So that'd be 2 out of 4, or 0.5. C, a colorblind son. We see right here that it's a quarter ratio, or 0.25 frequency of having a colorblind son. And D, having a colorblind daughter. None of the daughters of in the progeny are going to be colorblind, so it would be a 0 ratio or probability. All right, pedigree analysis. How do we read pedigrees? All right, so put simply, the squares right here represent a male, the circles represent a female. Now, if we have a line between both a male and a female, this signifies marriage or sexual intercourse. And this line going down from it signifies a line of descent. Meaning that these are the offspring, all these right here are the offspring that these two individuals produced. So the line that connects all of these individuals together signifies that they are siblings. And in this case, this male right here married a female from a different family, and they have another line of descent, and so on. That's how you essentially read a pedigree. Now you see these colored in portions signify that the individual carries a specific condition that we want to track within the family. All right, so first and foremost, let's read through the questions. Is it sex-linked? Is it dominant? Is it recessive? All right, so is it sex-linked? So for it to be sex-linked, and when people refer to sex-linked traits, they are mainly referring to traits located on the X chromosome. Unless it specifically says this is a Y-linked trait, always assume it's X-linked. All right, so for X-linked individuals, we see that we have a male that does not have the condition, and a female that does have the condition. If it was sex-linked, and we had a male with no condition and a female with the condition, for it to be sex-linked, it's going to be recessive, so she has to have 2 copies. That would mean that all of the females would not have this condition, yet all of the males would, because the females are inheriting one working X chromosome from their father. But we can see here that this female has the condition, so not sex-linked. Is it dominant? Here we see that we have 2 individuals not carrying the condition, yet they had an offspring that is carrying the condition. Would that occur for dominant individuals? Would this condition be dominant? No. It would not make any sense for 2 individuals who have a recessive phenotype to produce an individual with dominant traits or a dominant condition. That doesn't follow the patterns of inheritance. Is it recessive? Well, probably. It's following patterns of recessive inheritance. We have an individual here who does not have the condition, and an individual here who does have the condition. They had 2 offsprings who do have the condition. And the other side, we had 2 individuals that did not have the condition, and they are producing 1 offspring that does have the condition and 3 other offsprings that don't have the condition. So it is following recessive patterns of inheritance.